Good day everyone. I welcome you all to another practical pharmacognosy 1 session, that is PCG 212, and I, Mrs. Olale, am your lecturer for this practical schedule. For this practical schedule, we're looking at microscopy of crude vegetable drugs 2, with emphasis on transverse and longitudinal sectioning and histochemical techniques with respect to stem samples, and our stem sample of interest is water leaf, that's botanically known as Talinum triangulae. The outline for this schedule is as follows. Objectives: look at the objectives, the learning outcome, the introduction, sectioning stem specimen, microscopic evaluation of Talinum triangulae stem. Then I have also included the references and recommended textbooks. The objectives of this study is as follows. The objective of this study is to enable the student know how to cut sections of crude vegetable drugs, particularly the stem samples, using the transverse and longitudinal sectioning method. Second objective is to help the student know the microscopic features present in crude vegetable stem drugs, that is our Talinum triangulae that we'll be working with today through microscopic evaluation. The learning outcomes: at the end of this lecture, students should be able to understand how to cut transverse and longitudinal sections of specimens. Students should be able to evaluate crude vegetable drugs, particularly stem samples, using the microscope. And students should be able to draw sectoral diagrams of sections at times 10 and at times 40. Then students should also know some microscopic features in crude stem drugs. Introduction: cutting sections of specimen helps the students to understand the microscopic features that can be useful in identification of crude vegetable drugs. Transverse or longitudinal sections, they are usually employed in anatomical studies. You can use them to study stem samples, root samples, as well as leaf samples, but our emphasis for this schedule is on stem samples. So using appropriate staining reagents can also facilitate the identity of the structures that can be identified in some of these sections. So the figure below is a figure of water leaf plants, that is Talinum triangulae, showing the stems. The procedures for sectioning crude stem is given as follows. For the transverse sectioning, when you want to do a transverse sectioning of the stem sample, you hold the stem with your left hand. That is most when we are looking at most people that are always right-handed. You hold the stem in your left hand in a vertical position and use your blade to cut through the sample in a horizontal manner. That is, use your blade on your right hand to cut through the sample in a horizontal manner. For the longitudinal section, you hold the stem on your left hand in a vertical position as well, and you use your blade to cut through the sample in a vertical manner, that's going from up to down, vertical positioning. So this is what I was trying to explain earlier. This is the orientation of sections for the study of stem samples. The figure A shows the transverse section you cut in a horizontal manner, while figure B shows longitudinal section where you cut in a vertical manner. For histochemical techniques for microscopic evaluation of stem samples, this simply has to do with the procedures that needs to be taken, the steps that needs to be carried out in order to study the microscopic features present in stem samples. The first is you need to cut a thin section of your samples, either in a transverse or longitudinal section. And if you are provided with dry samples, you need to soften your samples before cutting them. Then you place your thin section that you have cut on a microscope slide. You clear the thin sections with chlorohydrate solution. Then you irrigate the cleared sample specimen with fluoglucinol stem. When I mean irrigate, just add few drops of fluoglucinol reagent. Then you leave for 2 minutes. After 2 minutes, you add hydrochloric acid. After adding hydrochloric acid, you wait for another 2 minutes. Then you carefully wash up the staining reagent. Please take care that when you're washing the staining reagent from your slide, you don't wash away your samples, your specimen, because it's going to be work done the procedure. You have to start all over again. So you have to take care when you're washing up the staining reagent that you don't wash away your specimen. After washing the staining reagents, you add a drop of your maltane. In the lab, we usually use glycerol. Then you cover with your cover slip. And when you're covering with cover slips, take care that you don't allow bubbles to be formed on your slide, because by the time you start mount under the microscope, you will just be seeing bubbles. under the microscope. So please take care when you are covering your samples with cover sleeve not to trap in some air on your slide. Then you view the specimen at different fields. So that means you move around your stage to view, because most of the time you can't see all the features in one location. You have to move to different fields of your stage, of your slide, and view your different features. So you view at times 10 and at times 40. So you present a representation of your image that will be seen under the microscope. And when you are presenting your representation, you have to give three figures. Remember, you give the magnification at which you are drawing, the eyepiece, and the objective length. Is very important. So the microscopic evaluation of phthalate triangulae, this simply has to do with the features that you see when you are observing your samples under the microscope. When you cut a transverse section of phthalate triangulae stem, you see that it's oval or circular in shape with a diameter of 5 to 10 mm. That means that's just the thickness. It's very thin. Very thin section is what you need. Then you see the vascular bundles. They are usually arranged 8 of them in number, and they are separated from one another. Then you also see the cortical region, which is divided into two different zones. You have the collenchyma and the parenchymal cells. Then you see the vessels. They are singlet too. They are solitary, thick walls, and usually occur in hexagonal shapes. Other features, you see the central pit region is wide and composed of thin wall round to polygonal parenchymal cells. Then you also see calcium oxalate. The calcium oxalate you see better, you see more after clearing the chlorhydrate. If you don't clear with chlorhydrate, you will not see your calcium oxalate. Then you also see the mucilage, which is contained in the parenchymal cells. The other features, you see the pericyclic fiber, the xylem, and the phloem fiber. So this is the pictorial diagram of the transverse section of phthalate triangulae. The features you see include, the first one is the epidermal cell. That is the epidermis. You see that's the outer part when you cut the section. Then you see the cortex, you see the collenchyma, you see the lysogenous cavity, and you also see the vascular bundles as well as the pith. All these features you will see when you observe under the microscope for phthalate triangulae stem. Then at a larger magnification, you see more features clearer. You see the pericyclic fibers, the phloem fibers, as well as the xylem fiber. That is at times 3. And at times 100, you see more of the vascular bundles being prominent at the higher magnification. So thank you. That is your references and your recommended textbook. Thank you for listening.